Okay, so this is looking at the triple topic of disease and defence. So the first kind of thing you need to know about is these three diseases, so HIV, AIDS, malaria and chlamydia. What causes them? This comes up quite a lot as like a 1-mark question, okay, so be careful of that. And again, bacteria, Trachomatis. Effect on infective organism, so different things that will affect to happen if you've got them, and then how we prevent it from spreading. Moving on down then to look at what monoclonal antibodies are. The mouse is vaccinated to start the formation of antibodies. Okay, antibodies for a mouse are made in the spleen, so you then basically take the spleen cells out in an operation. Okay, these spleen cells are then fused with tumour cells. Okay, and that forms something called a hybridoma. These hybridomas then undergo uncontrolled cell meiosis, and then the antibodies are collected. Uses of monoclonal antibodies: cancer cells, pregnancy test, blood test. Big thing now is they're used in COVID testing. Advantages of using them: monoclonal antibodies can be designed to bind and identify almost any substance. They can also treat conditions like cancer by carrying drugs directly to the tumour cells. Limitations: the human body is very complicated. Scientists originally thought monoclonal antibodies would be a magic bullet. However, this is not always the case. Some unwanted side effects: they're very expensive to produce, and then there's also the ethical issues of using mice. Why is it important that new drugs are tested? So we can work out whether they're toxic and if there's any other known side effects. This is also known as efficacy and checks how well drugs cure the disease and improve symptoms. There are 3 main stages, okay? So preclinical testing: drugs are tested on human cells and tissues. As we develop, they're tested a lot on computers as well now. Then we test them on living animals, and then we do healthy volunteers, okay? Start with really low doses and test on people to find the optimum dose, okay? Has to pass each stage. It can take up to 10 to 15 years for the whole process to occur. Advantages of testing drugs: new ways to cure diseases, improve the way doctors and nurses are trained, influence health laws and funding for health services. New treatment can cause serious side effects. New treatment may will work, or it may not be bettered as the standard treatment.